Hello friends, welcome to the Botany lecture series. The topic of today's discourse is Puccinia, its important features and life cycle. Puccinia is an obligate parasitic fungi causing rust disease in many plants. Let's first discuss its systematic position. Puccinia belongs to kingdom Myceti, division Amastigomycota, subdivision Basidiomycotina, class Basidiomycetes, subclass Teleomycetidae, order Eurydianales, family Pucciniaceae, and it belongs to the genus Puccinia. Puccinia is the largest genus of order Eurydianales, which causes rust disease of man's economic plants such as wheat, barley, oats, and so on. It is called rust because of the reddish-brown color of the spores that are found chiefly upon the surface of the host leaves and stems. The economically important species belonging to this genus include Puccinia graminis, the most important of all rust fungi, causes black stem rust of wheat. It occurs as an obligate parasite on a variety of hosts where it causes rust disease. Puccinia coronata, the cause of crown rust disease of oats. Puccinia hordi, the barley leaf rust. Puccinia sorghii, the common corn rust. Puccinia recondita, the wheat leaf rust. Puccinia stratiformis, the cause of stripe rust disease of various cereals, notably wheat and barley, and Puccinia iracidis, the peanut rust. Rust fungi may produce as many as 5 different stages in their life cycle. Stage 0, Eurydinia, bearing Eurydineospores, which are again dikaryotic. Stage 1, Telea, bearing teleospores, which are again dikaryotic. Stage 2 is Basidia, bearing basidiospores, which are uninucleate. Stage 3, Spermogonia, bearing Spermatia, which are haploid, and receptive hyphae, again uninucleate. Stage 4, Asia, bearing Asiospores, which are dikaryotic. Five stages will be discussed. Number 1, Eurydinia and Eurydineospores. The wheat plant becomes infected by means of Asiospores that are produced by the fungus on the alternate host leaf barberry. The hyphae produced by the Asiospores ramify through the host tissues, forming haustoria that draw nourishment from the surrounding cells of the wheat plant but do not kill them. In the early summer, the binucleate mycelium leads to the production of elongated, rust-colored, reddish-brown linear streaks or pustules on the leaves and stem. These streaks are known as eurydinia. From the base of each eurydium arise specialized hyphae, the sporophores, which bear at their apices oval, thick-walled, brown, unicellular, dikaryotic eurydeniospores. Due to them being produced in successive crops in a season, causing a widespread infection from plant to plant throughout the fields, the eurydeniospores are also called repeating spores. The rusty red color of eurydinia gives the characteristic name rust to the fungus and disease it causes. Each eurydeniospore has 4 germ pores around the equatorial plane. They are disseminated by wind currents to other wheat plants where they produce a new crop of eurydeniospores. The eurydeniospores germinate within 4 or 5 days by sending out a germ tube if sufficient moisture is present. The germ tube does not enter the host tissue directly. The germ tube grows over the surface of the epidermis, and on reaching a stoma, the tip swells up to form an elongated aprissorium from which the empty germ tube is cut off by a cross wall. The aprissorium gives rise to a fine branch that passes through the stoma and swells up to form substomatal vesicle. From this vesicle arise one or more hyphae which ramify through the intercellular spaces of the host tissue, producing a much branched mycelium made of binucleate cells. Now the second stage, telia and teliospores. In the late summer and early autumn, after the host plant matures, the same mycelium that gave rise to the eurydeniospores now begins to form another kind of spores, the teliospores. They are produced either in the same pustule that has been producing eurydeniospores or in new pustules called telia. These telia form elongated, dark brown to black pustules on the stems and leaves of the host plant. This is the black stage. The dark brown teliospores producing dark brown to black pustules on the surface of the stems and leaves of the wheat has given the common name black stem rust of wheat to this fungus. The teliospores are stalked, dark brown to black in color, thick-walled and 2-celled, each cell containing a pair of nuclei, that is, it is dikaryotic. In early spring, prior to germination, the 2 haploid nuclei of each cell fuse to form a diploid nucleus. Unlike eurydeniospores, the teliospores are not capable of immediate germination. They are overwintering spores which infect neither wheat nor barberry. Now the third stage, basidia and basidiospores. During the early spring, the teliospores germinate on the ground. Each of the teliospores forms a short, curved germ tube-like structure called a basidium. Basidium is also known as promycelium or epibasidium. The diploid nucleus in each teliospore cell migrates to the basidium, where it undergoes meiosis to form 4 haploid nuclei. These basidioforms cannot infect the wheat or any other member of the wheat family, but are capable to infect only the young leaves, fruits or twigs of the alternate host, that is the barberry. From the side of each of these 4 cells arise a short, narrow tube, the sterigma. The nucleus and most of the cytoplasm of each basidial cell migrates into its sterigma, on which a single haploid basidiospore is formed. Puccinia graminis is heterothallic, and out of the 4 basidiospores thus formed, 2 are of plus strain and the other 2 are of minus strain. Now comes the 4th stage, the spermagonia and spermatia. In the spring, each basidiospore germinates on the surface of the barberry leaf in the presence of moisture, forming a cylinder germ tube. The germ tube penetrates the epidermis directly and branches freely, forming a haploid monokaryotic, that is uninucleate intercellular mycelium, depending on the strain of basidiospores. The mycelium may be either of minus or of plus strain. Since many basidiospores infect the same barberry leaf, both plus and minus mycelia are developed side by side in the barberry tissues. After a few days of infection of the host, the monokaryotic mycelium branch and grow vigorously, producing network of uninucleate hyphae between the upper epidermis and palisade tissue of the host leaf. The hyphae towards the upper surfaces of leaves form spermagonial or pycnial primodia, whereas those towards lower surfaces of leaves form axial primodia. A few days after infection, the primodia of the spermagonia give rise to spermagonia or pycnia. The spermagonia may be plus or minus strain depending on the basidiospores from which they are derived. A mature spermagonium is an oval to flask-shaped pustule projecting from the surface of the leaf by a pore-like opening, the ostiole. The spermagonium is lined by cells that form sticky uninucleate cells called spermatia. The ostiole is surrounded by a bush of orange, stiff, unbranched, pointed hairs, the periphyses, which hold droplets of sugary, sweet-smelling nectar. The nectar, which is attractive to flies, contains the spermatia or pycniospores. The spermatia, which are produced in tremendous number, are finally exuded out and out of the spermagonial cavity through the ostiole. Among the periphyses, branched receptive hyphae are also found. Flies visit the spermagonia and feed on the nectar. While moving from one spermagonium to the another, they transfer spermatia to the compatible receptive hyphae. The receptive hyphae essentially act as the female gametangia, and on coming in contact with spermatia that function as male cells, bring about plasmogamy, and dikaryotic phase is thus produced. From the dikaryotic hyphae are produced axial initials towards the lower leaf surface. Ascia are thus formed on the lower surface of leaf from the dikaryotic hyphae. Now comes the 5th stage and final stage of Puccinia, ascia and asciospores. An ascium consists of a group of typically dikaryotic hyphal cells within the parasitized host, that is barberry, that gives rise to chains of dikaryotic spores called asciospores. Asciospores each containing a plus nucleus and a minus nucleus, are the first binucleate or dikaryotic spores produced in the life history of fungus. The achiospore chain eventually break through the lower epidermis of the leaf, permitting the spores to escape. The achiospores are now disseminated by the wind or at times by insects, and under favorable conditions they germinate and form a presoria. If germination occurs on a susceptible grass host, that is wheat, infection results and a dikaryotic mycelium develops. But if the achiospore germinates elsewhere, the germ tube develops only until all the stored food material in the spore is utilized and then dies. However, not every species produces all the 5 stages mentioned earlier. Based on the life cycle patterns, species of uridinales may be divided into 3 categories. Number 1, macrocyclic forms. Number 2, demicyclic forms. And number 3, microcyclic forms. Macrocyclic forms typically exhibit all the 5 reproductive stages, while demicyclic forms lack a uridinal stage. In microcyclic forms, both achiospores and urideneospores are absent, and the teleospore is the only binucleate spore produced. Although normally present in all the 3 life cycle forms mentioned earlier, spermagonia may be occasionally absent. All rust fungi, however, produce teleospores. Puccinia graminis is one of the most destructive and widely studied of all the rusts. The mycelium is well developed and consists of septate and intercellular hyphae, obtaining the nourishment by sending small, round or branched haustoria into the host cells. Puccinia graminis is heterophilic, macrocyclic, that is, it produces all the 5 reproductive stages during its life cycle, and it is heteroecious, that is, it requires 2 taxonomically different hosts to complete its life cycle. It cycles between wheat, Triticum vulgare, and barberry, Barbaris vulgaris. Stages 2 and 3, already discussed, are produced on the wheat, that is the primary host, and stages 0 and 1 on barbaris, that is the alternate host of the fungi. The different spore forms follow one another in a definite sequence within a span of 1 year. During the life cycle, 2 types of mycelia are produced, the monokaryotic in barberry and the dikaryotic mycelium in primary host, that is wheat. Now, let us discuss the control measures of rust disease. A tremendous amount of time and work have been put into efforts relating to the control of black stem rust disease of wheat. The methods of controlling the rust disease on the wheat plant can be summarized as: number 1, eradication of barberry, Barbaris vulgaris. The alternate host has shown a great deal of success in eliminating the black rust. The eradication of barberries also eliminates the possibility of recombination of genes and the production of new races, because sexual reproduction in Puccinia graminis takes place on barberry. However, in India, the eradication of barberry does not play any part in annual recurrence of the rust, as the rust survives in the uridineal stage on stray or self-sown wheat plants and other collateral hosts. Number 2, sulfur dusting at the rate of 30 pounds per acre at intervals of 4 days during infection period provides a good measure of control of the disease. Diethane cyclohexamide derivatives like Zainab, etc. may be successfully used. Early sowing of wheat may also prevent the rust outbreak. And lastly, the cultivation of resistant varieties of wheat is the most effective method of control. So friends, this was all about the important features and life cycle of Puccinia. Hope you might have understood. See you again in the next lecture. Till then, goodbye.